Hello and welcome. Today we will discuss estrogens and progestins used as drugs. This is one of the most important and clinically integrated topics in endocrine pharmacology because female reproductive hormones influence not only reproduction, but also bone metabolism, cardiovascular physiology, the central nervous system, coagulation, metabolism, and cancer biology. So, let's start. The learning objectives are shown. Please take a look. This lecture integrates physiology, endocrinology, oncology, contraception, infertility, menopause management, and receptor pharmacology. An important concept throughout this lecture is that estrogen and progesterone do not work independently. They continuously interact with one another through carefully regulated physiological cycles. Therefore, understanding the balance between these hormones is essential for understanding both physiology and therapeutics. Before discussing hormone pharmacology, we first need to understand the hypothalamo-pituitary-ovarian axis. GnRH released from the hypothalamus stimulates the anterior pituitary gland to release LH and FSH. LH primarily acts on theca cells, whereas FSH mainly acts on granulosa cells. Granulosa cells support follicular growth and estrogen synthesis. Progesterone and estradiol exert negative feedback inhibition on both the hypothalamus and pituitary gland. Notice the important regulatory role of inhibin, activin, and follistatin. Inhibin suppresses FSH release. Activin stimulates follicular growth and FSH secretion, effectively opening the FSH window. Follistatin neutralizes activin and supports inhibin activity. All steroid hormones originate from cholesterol. Notice the progressive reduction in carbon number from cholesterol to progestins, androgens, and estrogens. Progestins contain 21 carbons, androgens contain 19 carbons, and estrogens contain 18 carbons. This slide demonstrates the biochemical relationship between these hormone classes. Clinically, this relationship explains why some synthetic hormones may possess overlapping androgenic, estrogenic, or progestational effects. For example, some older progestins derived from testosterone may exhibit androgenic adverse effects. This structural relationship therefore has major therapeutic implications. An integrated endocrine cooperation is fundamental to female reproductive physiology, as shown here in terms of steroid biosynthesis. Theca cells mainly use the delta-5 pathway, whereas the corpus luteum primarily uses the delta-4 pathway. Cholesterol undergoes multiple enzymatic transformations leading to progesterone, androgens, and estrogens. Notice the central role of aromatase in converting androgens into estrogens. This enzyme is clinically extremely important because aromatase inhibitors are major drugs used in hormone-dependent breast cancer. This slide also highlights how different ovarian cell types cooperate in hormone synthesis. Theca cells generate androgenic precursors, which granulosa cells convert into estrogens under FSH stimulation. This slide summarizes classification of estrogenic and progestational drugs. Estrogens may be natural or synthetic. Estradiol is the principal natural estrogen, whereas ethinyl estradiol is a commonly used synthetic estrogen. Nonsteroidal estrogens such as diethylstilbestrol are structurally different but still activate estrogen receptors. Progestins may also be natural or synthetic. Natural progesterone belongs to the 21-carbon pregnane group. Some synthetic progestins are derived from testosterone and are classified as 19-nor compounds. Estranes and gonanes are important subclasses. Gonanes such as levonorgestrel and desogestrel generally possess lower androgenicity compared with older estranes. Estrogen and progesterone primarily act through intracellular nuclear receptors. Estrogen receptors include ER-alpha and ER-beta. Progesterone receptors include PRA and PRB after hormone binding. Receptor complexes interact with DNA and regulate gene transcription. These genomic effects are relatively delayed because they involve protein synthesis. However, estrogens and progesterone may also produce rapid non-genomic effects through membrane-associated receptors. Notice the role of G-protein-coupled receptors and MAP kinase signaling pathways. Estrogen and progesterone function together throughout reproductive life. Estrogen prepares the reproductive system for proliferation, follicular growth, and ovulation. Progesterone stabilizes the endometrium and supports implantation and pregnancy maintenance. Notice the consequences of deficiency. Estrogen deficiency may lead to osteoporosis and vaginal atrophy. Progesterone deficiency may impair implantation and produce irregular bleeding. Another important point is unopposed estrogen exposure. Estrogen excess without progesterone may cause endometrial hyperplasia and increase cancer risk. This concept is highly important in hormone replacement therapy. Estrogen generally promotes proliferation, growth, motility, and sperm-friendly reproductive conditions. Progesterone generally stabilizes tissues, supports implantation, and maintains pregnancy. Notice the opposite effects on cervical mucus. Estrogen produces thin, watery mucus favorable for sperm penetration. Progesterone produces thick, viscid mucus resistant to sperm passage. Similarly, estrogen promotes uterine growth and contractility, whereas progesterone suppresses uterine contractility. This explains why progesterone is essential for maintenance of pregnancy. Under estrogenic influence during the fertile phase, cervical mucus becomes clear, watery, elastic, and highly stretchable. This is known as the Spinbarkeit phenomenon. The mucus becomes sperm-friendly and facilitates fertility. Under progesterone influence after ovulation, cervical mucus gradually becomes thick and resistant to sperm penetration. Estrogen and progesterone exert effects throughout the body. In the hypothalamus and pituitary gland, estrogens regulate gonadotropin secretion and trigger the mid-cycle LH surge. In the central nervous system, estrogens influence mood and cognition, whereas progesterone produces sedative and anxiolytic effects through GABA modulation. Progesterone also increases basal body temperature after ovulation. In bone, estrogens inhibit osteoclast activity and preserve bone density. In the cardiovascular system, estrogens generally increase HDL and reduce LDL cholesterol. However, estrogens also increase hepatic synthesis of clotting factors, contributing to thromboembolic risk. Both estrogens and progestins produce important adverse effects. Estrogens are associated with carcinogenic potential involving breast and reproductive tissues. Diethylstilbestrol is historically important because it produced delayed type D adverse effects including reproductive tract malignancies in exposed offspring, daughters of mothers who used it. Estrogens may also produce nausea, vomiting, mastalgia, migraine, thromboembolic disease, and metabolic disturbances. Progesterone-related adverse effects include menstrual irregularities, metabolic effects, fluid retention, and possible carcinogenic concerns. One major therapeutic use of estrogen is hormone replacement therapy. This may be required in primary ovarian failure, premature menopause, or postmenopausal estrogen deficiency. Estrogens are also important components of hormonal contraceptives. Historically, estrogens were also used in testosterone-dependent prostate carcinoma, although GnRH analogs are now generally preferred. This slide highlights the remarkable diversity of estrogen pharmacology. The same hormone may be used for contraception, menopause treatment, or oncological purposes depending upon context. Progesterone and synthetic progestins are widely used in hormone replacement therapy and contraception. Notice the distinction between C21 compounds and 19-nor compounds. The gonadal subgroup generally has lower androgenicity and therefore better tolerability. Progestins are major components of modern hormonal contraceptives because they suppress ovulation, thicken cervical mucus, and alter the endometrium. When estrogen is given to postmenopausal women with an intact uterus, progesterone is added to protect against endometrial hyperplasia. To conclude, estrogen and progesterone pharmacology is deeply integrated with reproductive physiology, endocrine regulation, oncology, contraception, menopause management, and molecular receptor pharmacology. The most important concepts from this lecture include the HPO axis, tissue-specific receptor actions, and the balance between estrogen and progesterone. Thanks for watching. Don't forget to like, subscribe, and share. Happy learning!